Okay, so we'll discuss about transplantation immunology today. So, transplantation immunology exactly means all those immune reactions that are involved during the transplantation of the organs. And if you see, then kidney, pancreas, heart, lung, liver, bone marrow, and cornea transplants are performed in non-identical individuals. We'll come to what non-identical individuals are, and there is the ever-increasing frequency and success. Although the supply of organs is very less as compared to the persons who require it. And why do we require actually organ transplants is because of some kind of diseases or accidents. The organs, they undergo severe damage, and the individual can be saved if he or she gets a replacement for that particular organ. So that is why the organs are required. Now we'll go to the details one by one. So any tissue, any organ that is transplanted is known as a graft, and grafts can be classified into different types or have been classified into different types. Autologous grafts, they are also known as autografts. They are a graft transplanted from one part of the body to another in the same individuals. The most common type of this transplant is the skin transplant, where in case of certain burn accidents, the skin from the same individual, mostly from the hip region, is used is used as a graft and is implanted on the other part which requires the skin graft. So there are no chances of rejection in that case because of the fact that it is obtained from the same is being done. Then we have syngeneic grafts or isografts. These are grafts transplanted between two genetically identical individuals of the same species. So if there are genetically identical individuals available, then organs from them or the tissues from them can be taken and can be transplanted, and in this case the grafts face no chance of rejection. The grafts are accepted because of the genetical identity. This can be done in experimentally in case of mice, which are inbred strains of mice, also known as syngenic mice, which have the same genetic component. However, in case of humans, this is possible if there are monozygotic twins. It can happen between monozygotic twins because monozygotic twins are the only syngenic individuals available in humans, and in most of the cases they are not available. So we come to the third type of graft, which is the most common type of graft that is carried out, and this is allograft or allogenic grafts. The grafts transplanted between two genetically different individuals of the same species, like kidney transplants from non-identical individuals or non-syngenic individuals is one such example. And most of the transplantations or most of the graft transplants fall into this category of allografts. And then you have xenogenic grafts, or also known as xenografts, which are grafts transplanted between individuals of different species. That means heart of a baboon or heart of a chimpanzee if it is transplanted into humans. So this is a xenograft, and it faces the highest chances of rejection, whereas allografts are also most commonly can be rejected if not maintained properly. So these are the four different types of grafts, and if you remember, we discussed previously that the greater is the phylogenetic distance between the organisms, the more are the chances of uh, the proteins or the tissues being recognized as foreign in those individuals, and that it goes for the case of xenografts, and xenografts are most vigorously rejected. Okay, so if we go ahead, so you can see this this is an autograft within an individual, then isograft between identical twins, and then you have allograft which is non-identical, and then you have xenograft which is across the species, between different species it is done. Now we'll come to what is the basis of transplant rejection. So we have inbred strains of mice in which all genes are identical, also known as syngenic mice. So skin from an inbred mouse grafted into the same strain of the mouse is accepted, and there is no uh, rejection in this case. However, if skin from one inbred strain of mouse is grafted into a different strain of the mice, then it is generally rejected. So this is generally seen, and when we come to the uh, the basis of it, so between syngenic mice or inbred strains of mice who are genetically identical, the grafts are generally accepted, whereas in case of skin from one inbred strain, if it is transplanted into another strain of the mice, inbred strain of the mice, that is generally rejected. So what are the immune molecules that are responsible for it? Generally, they are okay. So we'll come to that later on. Then first we can see that primary rejection, and then we have two types of rejection. The secondary rejection. Okay, so what actually primary set of rejection and secondary set of rejection means? So this forms the basis of the immunologic specificity and memory. This is based on that. What happens is if you have two different inbred strains of mice and you implant or you transplant skin from one mice into another, what you will observe is primary set of rejection where skin gets primarily vascularized, and then finally the infiltration by different immune cells occurs and the skin is rejected within 10 days. However, if in the same mice the skin from the same inbred strain of mice which was used from the used in the previous experiment, if it is transplanted, then we see a secondary set of rejection. What happens in secondary set of rejection? The rejection is quite fast. It occurs within 3 days, whereas primary set of rejection occurs in 10 days. However, if this mice, the mice in which rejection has been carried out, if the skin is implanted from a different inbred strain of mice than that was done before, what we see is a primary set of rejection, clearly indicating that there is some basis of memory and specificity, that the the recipient mice, it actually develops a kind of memory develops memory for the previous exposure to the inbred strain of mice, and therefore a secondary set of rejection occurs. Whereas in case of a transplant from another set of another inbred strain of mice, what we see is a primary set of rejection, not a secondary set of rejection. So this is what is primary. See, this is the example which clearly indicates that if you do that, then the primary rejection occurs, whereas secondary rejection occurs in the first case. Okay. Now, transplant rejection is due to an antigen-specific response with immunologic memory. That is what is the basis of this, and this is what I wanted to tell you. Now, graft rejection, therefore, is a kind of specific immune response. It has specificity and immune memory. So, therefore, graft rejections are follow two types of pattern. One is first set of rejection if there is no previous exposure, and then another is second set of rejection if there is a previous exposure to the mice to the particular tissue. Okay, so if we talk about immunologic basis, then transplantation antigens, the major histocompatibility molecules or MHC molecules are the major players in this. That is, they are responsible for majority of the rejection mechanisms, and then we have minor histocompatibility antigens, and there are certain other alloantigens such as blood group antigens which are responsible for this. So major histocompatibility antigens, they are the main antigens of graft rejection. They cause fast and strong rejection, and there is difference of HLA types in the individuals, as you already know. We have talked about this in previous classes, and this is the main cause of human graft rejection because HLA alleles are quite diverse and they are not same between the individuals. So that is why the set of grafts allografts show rejection. Minor histocompatibility antigens, they are also cause graft rejection, but they are very slow and weak. And then this is what you see is a skin graft to a syngenic recipient, which means graft is tolerated because there is no difference of MHC antigens, whereas in case of allogenic recipient, there is difference between uh, the between the genetic component, genetic constitution, as well as we refer here particularly to MHC. And if you see, the graft gets rejected uh, very rapidly if you have the response to MHC, whereas in case of minor histocompatibility antigens, the response is slow and the graft is rejected very slowly and can be managed easily by using certain other therapies. Okay. So human ABO blood group antigens are the other type of alloantigens against which response can develop, and therefore rejection can occur. And there are certain tissue specific antigens that can also cause this kind of rejections. Okay, so talking about mechanisms of allograft rejection, there are many mechanisms. Cell-mediated immunity is responsible, humoral immunity is responsible, and NK cells are also responsible for these kind of things. Okay, so I think I'll stop here and then we'll continue with it in the next lecture.